All right, next up we want to talk about DNA technologies, starting off with DNA sequencing. DNA sequencing is just determining the order of nucleotides in DNA. This is going to allow researchers to identify alleles that are associated with a disease so that way they can develop treatments to target those particular alleles. This is also going to aid in forensic biology and contribute to the understanding of our evolutionary history. So to begin sequencing a segment of DNA, many copies of the segment are going to be made using a special technique called polymerase chain reaction. So a polymerase chain reaction, or if you go on to take many other courses, you're going to see this frequently abbreviated to just PCR. This is going to use DNA polymerase to copy a segment of DNA. And it's not going to just copy it once or twice, it's going to copy it hundreds and hundreds and thousands of times. It's going to involve three basic steps. First is denaturation. Denaturation in DNA is going to occur much like denaturation in proteins. Denaturation breaks those hydrogen bonds that are holding the two strands of DNA together. Now, since the rest of the DNA strand is going to be covalently bonded, it's not going to be affected by the heat. Really, the only thing that's going to happen is the two strands of DNA are going to separate, just like they would with DNA helicase, only instead of breaking one hydrogen bond at a time, we're going to break all the hydrogen bonds at once. So we're going to heat up the DNA until it becomes single-stranded. Then we're going to begin the annealing process. During the annealing process, DNA is going to be slowly cooled down, and we're going to use a primer to help attach brand new nucleotides to each of the DNA strands. Extinction is going to be when we use DNA polymerase to add the complementary base pairs to each of the single DNA strands, and it's going to create two double-stranded DNAs. Once we have two copies of DNA where once there was one copy of DNA, we're going to repeat the process. We're going to denature the DNA, begin the annealing process with the primer, extend across the entire length of the DNA strand, and now we will have 4 DNA strands where once we had 2 DNA strands. Here's an example of what that looks like. So we start off with our single strand of our double-stranded DNA sequence. What happens is during the annealing process, or during the denaturation process, we are going to use heat to unwind and separate those two strands. When we separate those two strands, we end up with 2 single strands of DNA. DNA primer binds to that process, or binds to that those single strands, and then DNA polymerase can zip along and fill in the complementary nucleotide base pairs. Once it is completed, we now have 4 strands where once there were 2 strands. A second round begins, and we're going to end up with 8 copies of DNA. So after only a few rounds, you can see that it grows pretty exponentially. So after our first cycle, we have 2. After our second cycle, we have 4. After our third cycle, we have 8. After the fourth cycle, we have 16, and then 32, and then 64, and it's going to escalate really rapidly. So after, 100 cycles, we're going to have substantially more DNA. This is going to be used a lot when we are studying DNA. You're going to see it a lot for DNA fingerprinting or DNA profiling. This is why very little DNA is needed for forensics procedures. How you can take just a drop of spit and you can use a PCR polymerase chain reaction to produce as many copies of that DNA as you need to study it for whatever analysis that you need. So DNA fingerprinting or DNA profiling, we're going to use PCR to amplify fragments of that DNA and then gel electrophoresis to separate those fragments by size. If 2 DNA patterns match, they probably came from the same source. There are multiple uses for DNA fingerprinting. You can use it to test for viral infection, genetic disorders, cancers. You can use it to compare blood or tissue samples from a crime scene to criminals. You can use it to identify the remains of bodies for forensic medicines or identify victims of natural disasters. So there are a lot of uses for DNA fingerprinting. Here's what a gel electrophoresis would look like. You're going to take a sample of DNA from a crime scene, and you can take a sample of DNA from a suspect A and suspect B. After undergoing PCR, you can amplify that DNA so you have multiple copies. You then take that DNA and you can run it through a gel. When you run it through a gel using gel electrophoresis, you're going to separate out the different fragments of DNA. That banding pattern of how that DNA separates out is going to be unique to every individual. This is why we call it DNA fingerprinting. It's going to be as unique to an individual as their fingerprint. So when this banding pattern separates out, we're going to see that the DNA collected from suspect A has one band right around the 12 repeat mark. Suspect B has a band right around the 16 repeat mark and the 12 repeat mark. The DNA collected at the crime scene has a band at the 16 mark and at the 12 mark. So which suspect matches the DNA that was picked up at the crime scene? This region over here is the marker or the DNA ladder. This is necessary for every gel that is run. Anytime you run a gel, you must always have a ladder, a marker that's going to help indicate the exact location of different types of fragments. This is how you're able to measure and compare your DNA fragments. So, yes, which of these suspects matches the DNA that was found at the crime scene? So that's one that's one type of DNA technology. Another type of DNA technology or term that we want to use is cloning. Cloning is the production of genetically identical copies of DNA, cells, or organisms. There are lots of different types of cloning. Bacteria, by their very nature, clone themselves. This is how they are able to reproduce asexually. Cloning does happen a lot in the natural world. The process of twins, identical twins, are in essence natural clones of each other. They're two individuals with the exact same genetic information, the same DNA. So that would be cloning of whole organisms. We can see cloning of individual cells. That would happen at the cellular level, like in the case of bacterial reproduction. But we can also see the cloning of DNA sequences. So we're going to start off by talking about gene cloning. This is when we're producing multiple copies of a single gene. A lot of times we're going to do this in the case of recombinant DNA. Recombinant DNA contains DNA from multiple sources. Now this is going to be very important. We'll talk about why. But we're going to see recombinant DNA as a very important DNA technology. A great example of this would be the production of human insulin. Human insulin is a protein. Proteins are very large structurally complex. If you remember when we talked about the structure of the structure of proteins, we talked about the primary structure, which is the sequence of amino acids, the secondary structure, which would be those alpha helices and beta pleated sheets. Then we talked about the tertiary structure, which is the three-dimensional structure, and then potentially the quaternary structure, when we have multi-polypeptides that are bound up together, working together to form a single protein. Proteins, all that to say, proteins are very complex and very difficult, very expensive, and very time-consuming to create in a lab. However, bacteria are filled to the brim with wonderful, perfect little protein factories. Does anybody know what we call those protein factories? Ribosomes. What we have found is that if we give the genetic information, if we give the instructions for how to make human insulin to a bacteria, they can take that instructions, that information, and they can produce massive quantities of that protein insulin very rapidly, very efficiently, and very quickly, very easily for us to extract, purify, and then use. So recombinant DNA is where we're going to take the instructions for how to make human insulin, and then we're going to inject that into a bacteria. We're going to combine that with the bacteria's own DNA, and then use that bacteria to produce our gene of interest, or our gene product of interest. So we could do this if we just want to make a lot of copies of a particular gene, or we can do this if we want to make a lot of copies of a particular gene product, whatever protein is coming out of that gene. So to create recombinant DNA, a vector is going to be used to introduce the gene of interest into a host cell. Usually the host cell will be a bacterium. A common vector that we'd like to use is a plasmid. If you remember way on back when we talked about bacterial structure, we talked about how DNA or bacteria often have extra chromosomal DNA known as plasmids. These plasmids are little rings of DNA that are not part of the basic chromosome, but they do carry very important information that bacteria can pass back and forth. A lot of times the bacteria will pick up information like maybe the antibiotic resistance to a particular antibiotic. They keep that information in a plasmid, which they can then copy, replicate, and pass back and forth to their neighbors. Since bacteria are already doing this, they're already picking up plasmids, sharing plasmids, they're used to picking up DNA, we're going to use them as our hosts to pick up plasmids that we have created, artificial plasmids, small rings of DNA in a bacteria that aren't part of the bacterial chromosome. So here's how we're going to do it. First, we're going to use a restriction enzyme to cleave the human and the plasmid DNA. So here we have a human chromosome. We're going to use a restriction enzyme to cut here and here. That's going to be the gene for insulin. We're also going to use that same restriction enzyme to cut the plasmid. The plasmid is going to cut here and here. And when it cuts in those locations, it's going to fit in the puzzle piece of the human insulin gene that we want to insert in there. So restriction enzymes are pretty interesting. They do occur naturally in bacteria. Restriction enzymes are not something we've created or come up with. They occur naturally in bacteria. They're used to cut up viral DNA that enters the cell. So really, it's a defense mechanism against foreign DNA. We use these during cloning. It's going to create a gap in the plasmid DNA into which the foreign DNA can be placed. So we're going to cut open the plasmid, and then we're going to use DNA ligase. Remember, ligase is used to seal up those Okazaki fragments that are produced in DNA synthesis. We're going to use DNA ligase to seal in our gene of interest into the vector plasmid. We're going to use the same restriction enzyme to cleave both the human and the plasmid DNA. And the reason why we use one restriction enzyme to cleave both the human cell and the plasmid is because of when a restriction enzyme cleaves the DNA, it doesn't cleave it, it doesn't make a blunt end. It doesn't cut it right down here. Instead, it creates a jagged cut that's known as a sticky end. These sticky ends are going to be perfectly matching. So it's kind of like cutting a unique puzzle piece. So we're going to cut a unique puzzle piece on the human DNA, and we're going to cut that exact same shape in our vector. Or maybe it's more like Velcro. And what happens is that human DNA is going to have the perfect sticky end that's going to match this overhanging bit right here, and is also going to match the sequence of this overhanging bit right here. And it's going to kind of Velcro right into place. DNA ligase can then seal it in. So then we're going to introduce that DNA to the bacterial cells. Some of those bacterial cells are going to take up that recombinant plasmid that we created. Once they take that up, they're then going to clone that. They're going to just naturally clone that DNA over and over and over again. Once they've cloned that DNA, they are going to take that DNA, bring it to their ribosomes, and their ribosomes are going to start pumping out mass quantities of that gene product that they couldn't make before. Once they make that product, in this case human insulin, we can then isolate that insulin from the bacteria. We can harvest it, purify it, and then use it to give to patients who are lacking insulin, specifically patients suffering from diabetes and the inability to produce insulin of their own. All right, so that's, we're going to pause right there for our, we're going to do one more slide real quick. So for a human gene to be expressed in bacteria, it has to have bacterial regulatory regions. So that means that it's going to have it's not going to be able to contain introns. Bacteria don't have introns, so that's going to have to be removed before we can cleave that DNA.